Welcome back. This is part 2 of the first genetics lecture, and we're going to cover chromosomal patterns of inheritance. And what we mean by this is when we first started all this, we talked about Mendel and his mysterious factors, right? So the idea really was, if we think about the history of all this, we start out with the idea that two organisms come together and they form some kind of blend. So they just pretend, straight line, wavy line, and you get really slightly wavy line, right? So this is the idea of blending, okay? What Mendel then did was he took this concept and he said, no, instead what's really going on is organisms have some kind of discrete factors that they pass on to their offspring, okay? And the idea is that there's two factors for a given trait, okay? When you make gametes, each gamete just gets a single factor, and then these come back together again during fertilization, okay? The thing is so if we talk about these things, these factors, okay, and for the longest time in the early literature they called them Mendel's factors because they realized he was the first one to really clarify this and publish. So the question was, what on earth were Mendel's factors, right? And everybody had their own ideas. Darwin even had one where he called these things gemules. If you read up on Darwin, you can learn about something called his parthenogenesis theory. He was just completely wrong, but this was his attempt at developing what you would call a particulate theory of inheritance, okay, which would be distinct from a blending theory, right? So obviously today we know that genes are found on chromosomes, right? And that's not a good chromosome. Here's a good chromosome. Hold on. Okay, we got that, and then we got our little centromere. Okay, so there's a chromosome. And chromosomes have bands that you can find when you stain them. And if you zoom in, right, you zoom all the way in, you can get to the double-stranded DNA helix. And genes are basically just sequences of the A, T, T, C, G, A. I'm just making this up, right? There's the A, the T, the C, and the G, right? You guys remember all this from an earlier chapter. So what we're going to do now is we're going to make the jump from Mendel's mysterious factors that we had no idea what they were, and we're going to start talking about what we know today, which is the fact that those factors are actually genes located on chromosomes. Okay? So here we go. So let's make sure we understand our own human genome's basic organization. So you can take our entire genome, right, the entire complement of all of our DNA, and you can cut it up into a bunch of pieces. The first 22 pieces we're going to call the autosomes, and the second 2 pieces we're going to talk about are the sex chromosomes. Okay, so you have the autosomes and you have the sex chromosomes, and together that makes up our entire genome. One way to think of it is if you take all the DNA in a given cell, okay, a single cell has about 6 feet of DNA, okay? The diameter is about 2 nanometers. Okay, now just think about this. A single cell in its nucleus, which is about as large possibly as 100 microns, okay, micrometer is equal to, so this would be 100, this would be 100 over 1,000, or 1/10 of a millimeter. Okay, that's how small this is. So we have basically 2 meters of DNA wrapped up into a space that is less than a hair's width thick. Okay, that's a little ridiculous. So what we can do is imagine taking that big long chain and then just cutting it into random pieces, okay? And so now you can imagine each one of these chunks, that's one of those pieces, okay? If you add all these together, including the X and the Y, of course, you get what is essentially, like a dotted line, and you call that your genome, okay? There is no rhyme or reason to how many chromosomes an individual species has. there's no relationship to the fact that we have 22 while, say, fruit flies have 4, or peas have 16 and mice actually have 16. There's no relationship between the number of chromosomes and the complexity of the individual, or the complexity of the species. Okay, it's just the way it is. So we talked earlier about, in humans, we have sex-linked traits, right? And so chromosomes themselves can be represented in the same way that genes are, with a single letter. So for example, you can say the X chromosome, the Y chromosome, okay? And so we know that in mammals, XY is male and XX is female. And we care about genes on the X chromosome because males only have a single copy of them, and so therefore a single factor or allele, while females have 2. Okay, so this leads to interesting genetic phenomena. It should be noted that the Y chromosome, even though there are genes on the Y chromosome has fewer than 100 protein coding genes. Okay, and over evolutionary time, the Y chromosome has kind of been evaporating slowly. Well, that's another story for another time. All right, so we call traits associated with genes on both the X and Y chromosomes sex-linked genes. And because X chromosomes are bigger, and therefore there's a lot more genes, many of the sex-linked genes we learn about are located on the X chromosome. So we therefore call these X-linked genes, which of course are associated with X-linked alleles. Okay, so here we notice that males only have a single allele for a gene pointed out in red. Okay, so for mom, it's just like any other gene. She could be dominant or recessive, but dad's different. He only has a single allele for the gene. So we would call this state being either homozygous or heterozygous. But this state is called hemizygous. Okay? All right. So that means if dad has a recessive allele, he will express its phenotype. There's just no compensating for the recessive trait with a dominant allele. So dad can never carry a recessive trait on the X chromosome. He either has it or he doesn't. All right, so let's use this as an example. We're going to use fruit flies. So there was a guy in the early 1900s, and in 1912, actually, he published an article that showed and demonstrated the linkage between a phenotype in a fly and the pattern of inheritance of the X chromosome. So Thomas Morgan was the first person to say, hey, those mysterious factors of Mendel, they are actually physically located on chromosomes based on the patterns we see in our experiments. All right, so let's kind of revisit his experiment. So let's say that capital R stands for red eyes and lowercase r stands for white eyes. Okay, so because we know the gene for eye color is on the X chromosome, we can use the following notation, right? So you do like a superscript for the X. Okay, and by the way, the first mutant fly that was ever discovered actually had white eyes, so that's why we can say that white's recessive, right? So up until Thomas Morgan discovered the white-eyed male fly in his lab, red eyes were the norm. Okay, so we would call red eyes the wild type. Okay, and the white eyes would be the mutant. Okay, so a white-eyed fly or a red-eyed fly, we can signify the alleles in the slightly more complicated way, saying that there's this allele R, capital R, lowercase r, and it's located on the X chromosome. Okay, so you can see there's the capital R allele, okay, found on the X chromosome. Okay, now let's tell the story of Morgan's first mutant fly. So Thomas Morgan was investigating genetics or inheritance in fruit flies, trying to repeat some of Mendel's work. And unlike Mendel, who could just go to market and find a bunch of peas and different varieties of them, Morgan didn't have a fly market to go to and find a whole bunch of different varieties of fruit flies. And so he had to find them himself. And so it took him years of staring under microscopes and looking at flies until one day they finally found the first fly that instead of having red eyes, it had white eyes. And so they took that white-eyed male and they crossed it with the red-eyed female. And sure enough, all those F1 progeny had red eyes. Okay, and so they got excited. They saw the dominant recessive relationship Mendel saw in peas. And then they did the next thing. They did the F1 cross to F1. So they took a male and a female from that generation. They crossed them. And that's where we're going to start. Okay, so we have the F2s. Okay, the F2s, again, just like Mendel, gave us a 3-to-1 ratio of the red to the white. Okay, now you would think this is kind of boring and just confirms the experiment, but here's the thing. When they looked at all the white-eyed flies, they found that they were 100% male. Okay, they were 100% male, which violated Mendel's law, because when Mendel did his experiments, if he crossed, say, the yellow pea plant and the green pea plant, it didn't matter what the genders of the parents were. It was always the same. Okay, so what was going on here? Well, they hypothesized that the gene, or the factor that controls eye color, resides somehow, is associated with the X chromosome. Okay, so let's model that. So here's a female crossed with a male. Okay, and here's a wild type female, and here is the mutant male. Okay, and so when you do this cross, so this is the parental generation, when you do this cross, you get females that are hybrid, okay, red eyes because they have that extra allele, and males that are 100% red-eyed. Now you might be like, well, wait, why are all the males 100%? Because there's this here. Why isn't there a possibility that the male can inherit that? Well, remember, in order to be male, you have to have both an X and a Y, right? And so the only place you can get a Y chromosome is from dad, right? So all males actually have a maternal copy of the X chromosome. And because mom only had red eyes, she's going to only pass on an X chromosome with a capital R. Okay, so again, that F1 progeny was, the F1 progeny was hybrid. Okay, and so we're now going to take that F1 and cross it. So here is an F1 female. She is now carrying the allele for white eyes, although not expressing it herself. And here's the wild type male because he has red eyes. Okay, now when we set this cross up, okay, so there's mom, here's dad, we will note, okay, I'm just going to skip all the X's and XY's here just to write this out quick here. And you see that null, that indicates that they received the Y, right? So the Y chromosome doesn't have an allele for this, right? So really this is just a shorthand of writing that. Okay, so the F2 generation had a 3-to-1 ratio of red to white. But what was interesting here is that 100% of the males were white-eyed and 0% of the females were white-eyed. Okay, I'm sorry, not 100% of the white-eyed flies, rather, were male. 50% of the males were white-eyed, but of all the white-eyed flies they found, only the males were there. Okay, and so this was a violation of Mendel's laws, and exciting. So they hypothesized, as I said earlier, that the factor is on the X chromosome. So basically the theory here, or the model, fits the data. Okay, and so here's what I want you to do now. I want you to take this cross, okay, and I want you to figure out what we need up here in order to generate the first female with white eyes. Okay, so I'm going to let you pause it and think that through. Okay, so if you thought this through, you would be right that the dad has to have a lowercase r, okay, and mom has to be a carrier. Why? Because you have to get 2 X chromosomes, each with a lowercase r from a parent. Okay, and so of course Morgan's group did that, and sure enough they did find the female white-eyed flies. So this established for the first time that on chromosomes, genes are associated with them. And they knew that because a male has an X and a Y, and a female has an X and an X, and the only way that the reality fits the data of Morgan's experiments is if the factor, Mendel's factor for the gene, was in fact located on a chromosome. Okay? Morgan went on to win Nobel Prizes for this work. All right, now let's think about 2 traits. So 2 trait crosses also gave unexpected results that deviated from Mendel's laws when Morgan did them. So traits would actually travel together as a group, and so we can call this a linkage group. So a linkage group is a group of genes that are located in the same physical area of a chromosome. This large region we call a locus. And a linkage map shows the relative distance along the chromosome where the genes are located. So we can think of a chromosome, okay, and we can have multiple genes on the chromosome. And so let's just think about two genes, right? Let's just think about an A and a B, okay? And so you can just imagine here, if we have a pair of homologs, and let's say this person is heterozygous, and so they have a lowercase and an uppercase, all right? So the genotype here would be this. Okay, you can imagine during meiosis, this chromosome is going to go this way, and this chromosome is going to go that way. So this is always going to be capital, capital, and this will always be lowercase, lowercase. Okay, so that's what we mean by linkage. You would say that the A and B genes, or the A and B loci, are linked. So what you're seeing here is just a portion of one of Morgan's chromosome maps. Okay, there's a lot to take in here, so let's just go through this. At the top here, you can see here the wild type characteristics of the different traits that they observed and the subsequent mutant characteristics. So for example, there's the long antenna phenotype, and then that correlates with a short antenna phenotype. Okay, so when they first discovered this, for example, they crossed long antennaed with short antennaed, and they got long antennaed. And then they crossed those long antennaed hybrids, and they found a 3-to-1 ratio of short antenna, right? So for each one of these traits, they found a corresponding Mendelian relationship. Okay? So here, let's just, for our purposes, let's just label these first few here, A, B, C, D, E, F, G, H, right? And so these alleles would be the mutant recessive ones, okay? So you can imagine taking a fly that's, say, AACC, okay? And then doing that cross. Okay. Now, if this was traditional Mendelian inheritance, you would expect that familiar 9:3 to 3-to-1 ratio of that dihybrid cross. When Morgan started doing these crosses, though, he did not find that ratio. And instead what he found was for any given combination of dihybrids, so we just did A and C, but if he did, say, B and F, or if he did D and A, or A and D, they always found different ratios. I'm sure it drove them crazy when they first discovered this. But over time, what they realized, what was going on was that occasionally, even though these were on the same chromosome, they would occasionally be unlinked. And we understand this because of crossing over that occurs. Okay, so let's go forward for a minute. I'm going to re-explain crossing over to you. So here we have a pair of homologs. Okay, so there's a sister chromatid, there's a sister chromatid, there's a sister, there's a sister. Okay, this individual homolog is all homozygous. Contains all the capital alleles, and this one contains the lowercase alleles. Okay, and so you can now imagine what happens during crossing over, which what we call synapsis, when the non-sister chromatids line up. Okay, and so you can see that characterized over here. So here we go. We're going to just consider one pair of the non-sisters. Okay, so one red and one blue over there. Okay, and so what's happening when we do crossing over is someplace on this chromosome, some random place, enzymes pick, and so let's just pick in between A and B, and they end up cutting the DNA, and then exchanging information. Okay, I probably shouldn't have used red for this, but here, let's do green. Right, so we can do crossing over here. So this gets cut off, this gets cut, this gets cut. And when that gets cut, it gets pasted on to this area. And when this gets cut, it gets pasted on to that area. Okay, and so we can look at it like this. So basically, one chunk of a chromosome is now found on another. They traded places like musical chairs. Okay, and the key here is you now have a unique combination of alleles that have never been seen before. A lowercase a has never been on a chromosome with capital B, and then C and D, right? So that would be the genotype for any gamete getting that chromatid as a chromosome. Okay, and so you can see now we have 4 different kinds of genotypes. We have A, B, C, D capitals, right? We have lowercase a and then B, C, D. We have capital A, lowercase b, c, d, and we have lowercase a, b, c, d. Okay, so all four of these are now possible. We can see this increases genetic diversity. That's the whole point of this. All right, now we call any gamete that gets either of these chromosomes the parentals. Okay, the parental, because they have the original genome, the original combination, the original genotype of the parents. And we call these the recombinant alleles. And we call them recombinant because they have pieces of each. Okay, so if we go back now to here, see these numbers? Let me switch back to red. Okay, you see these numbers? These numbers correspond to the probability of crossing over occurring between these two loci on a chromosome, right? So if I took this blue bar, this chromosome, and I drew it like this, upside down, or along the side here, okay? And I took that first gene, and I took that second gene, okay? So the one that says 0 and 13, okay? So let's say that, again, the long antenna, let's say that this is the A gene, and this is the B gene, okay? The chances that crossing over will occur between those is 13%. Okay, and so what you can see here are map units. These are map units. And map units, one map unit is equal to a 1% chance of crossing over to occur. Okay, how did they arrive at those numbers? They did the experiments. Okay, they crossed a long antennaed, long-winged fly to a short antennaed, dumpy-winged fly. And they found that 13% of the offspring showed the recombinant phenotype. Okay, so now think about that for a second. You tell me, stop and think, what would the parental phenotype be? And what would be the recombinant phenotypes? Think about that. Okay, so maybe you paused it, maybe you didn't. If you didn't pause it and think, I would say pause it and really think hard about this, if you can figure this out. Okay, so the parental phenotype would be long antenna, long-winged, okay, or short antenna, dumpy-winged. The recombinant phenotype would be long antenna, dumpy-winged, or short antenna, long-winged, okay? And so you would see, if you counted all those flies up in that dihybrid cross, okay, if you did this kind of cross and you cross that to itself, you would see 13% of the flies that are long antenna, dumpy-winged, or short antenna, long-winged. Okay, how am I finding these? Long, dumpy, okay, so there's a cross here. This is the crossing over. Short antenna, long-winged, okay? That 13% is either of these, right? Because think about it. If this crossing over occurs, for every one of these, you get one of these, right? And so when you count up all the recombinant flies, you get 13%. And now you can imagine an experiment where you cross this one and you get 31%, right? You can imagine crossing this one and getting the difference between these numbers, okay? And you can do basically any combination so that you can eventually, when you take all those data and you put them all on paper and you do the math, you can now say that the gene for this is 13 map units away. Sorry, the gene for antenna length is 13 map units away from the wing gene, is 31 units away from the leg gene, etc., etc. These are relative distances. Okay, this would be the equivalent of saying, I live here and I live, twice as far away. So this is me. I live twice as far away from B as I do C. Okay, but how far is that? Is it miles? Is it inches? Who knows? The idea is it's relative. The key is that Morgan was able to say genes are on chromosomes and they're in physical spots that do not move. He found a home for Mendel's factors. That's the key here. Okay. Now, I'm never going to ask you how to solve mapping questions. What you're going to need to be able to do is understand and define the concept of linkage and discuss the importance of those crossing over frequencies, which in the book might call them recombination frequencies. Okay, so recombination is simply the more general term we use to describe when two pieces of DNA end up exchanging pieces. Okay, that's recombination. So understand the importance of these recombination frequencies to the localization of genes on the chromosomes, and identify why linked genes do not follow the law of independent assortment. Right. The key to understanding mapping is to understand that crossing over during meiosis. Okay? Okay, now I just went through all this, so I'm going to skip these slides. All right, moving on. We can now talk about very general, large-scale chromosomal mutations. Okay, and so let's get a few vocabulary words out of the way here. So when we talk about polyploidy, right, that's the extreme version of going from haploid, right? If you remember, a haploid cell has a 1N DNA content. So an egg for humans, right? If humans have an N equal to 23, right? You would say that therefore the eggs and sperm have 23 chromosomes. So that's, remember all that. Okay, so haploid, and then our somatic cells, our body cells, are diploid, right? And so that's a 2N content. But let's go further. Imagine an organism that is triploid, okay? And so they have a 3N content. So the concept of polyploidy means many copies, many sets of the entire genome, okay? Most plants are polyploid, okay? For example, corn and wheat, two of the most common grains that we grow. These have, I think, one is 6N, and I think wheat is octoploid, 8N, okay? All right, euploidy just means the norm. So in other words, a cell that is euploid means it has the correct number of chromosomes. All's well. It's a very general word, okay? Aneuploid means something's wrong. That's all it means. Okay, it can mean many different things. So here's the key though. 100% of cancer cells are aneuploid. By the time a cell has become cancer, it is so far gone from its roots of its original genome that it is completely an alien cell. What we call these aneuploid. All right, so there's many kinds of aneuploidy. When we talked earlier about, say, Down syndrome, right? Down syndrome was trisomy 21. That's aneuploid. Okay, we talked about Cry-du-chat. Okay, that's that loss of the tip of, if this is right, if this is chromosome 5, losing the tip of that, that's aneuploid. Okay, Turner syndrome. Turner syndrome is when you just have an X chromosome. Okay, Klinefelter is when you have multiple copies of the X chromosome. Okay, you have 47XYY, that's super male. That has an extra chromosome. All of those are conditions we call, those are all aneuploid conditions. Okay? All right. Now, how do we get there? How do we get trisomy or monosomy? How do we go from having a normal complement of chromosomes to an abnormal amount? One of the most common ways is a concept called nondisjunction. Okay? Nondisjunction is when, during meiosis, chromosomes inappropriately segregate. And this results in both either monosomies or trisomies, as we talk about in Down syndrome. So here's how it works. You can take a normal cell that goes through meiosis. Okay, so during meiosis 1, it divides in half. Okay, and you can see here, here's just a single copy of homologs, and you can see that the homologs separate, right, just like they're supposed to. In this cell, you can see that the sisters now separate. Okay, and so these two cells are normal, and when they get fertilized by an egg, by a sperm coming in, you can see that they form completely normal zygotes, right? A 2N content of zygotes. However, on this side, this pair of sisters does not segregate appropriately. It turns out both of them go to a single cell, and so one cell gets 2 and one cell gets none. this cell that gets 2 chromosomes. And again, this is just a single copy. Let's say this is chromosome 21. So over here that you're not seeing are all the other sets of chromosomes. Okay, so this cell doesn't have nothing. This cell simply lacks that chromosome. Whereas this cell has 2 of those chromosomes. Okay, so again, the sperm comes in, we get fertilization, and now we have an embryo, or rather a zygote, that has a normal complement of chromosomes for everything, except it has 1 extra for that chromosome. And likewise, for this cell, if it gets fertilized, it will only have a single copy, the copy that came in from the sperm. So it's a normal cell, but otherwise missing an entire chromosome. Okay, and if you recall, in humans, the only viable monosomy, so this would be a cell that is monosomy for that chromosome, the only viable monosomy is Turner syndrome, where you just have a single copy of X. So if this happens for chromosome 1 through 22, that does not result in a zygote that can move on and differentiate into develop into a fetus. Okay? Whereas there are a few of these trisomies, the most common being trisomy 21. All right, so this is what happens in meiosis 2. However, there are instances, more rare but far worse, where non-disjunction occurs in meiosis 1. And this is when the bivalent, or the tetrad, the homologous pair, fails to segregate, and so one cell gets both of them. And so you can see here, there are no normal cells, okay? All of them are either +1, so falling into a trisomy condition, or a -1, okay? Now, female mammals are born with all of the eggs they'll ever have, and all of those eggs are arrested in meiosis 1. I should say here, here are the eggs. They're arrested in meiosis 1, okay, and over time, they sit there and they age, and they get exposed to radiation and oxidative damage and all sorts of things. So as you get older and older and older, the chances of this occurring gets greater and greater and greater. This is why women who have children in their late 30s, early 40s, mid 40s have an increased chance of developing or having a non-disjunction event occur, resulting in a chromosomal abnormality for their fetus. Okay. This non-disjunction occurs during development, and that's why I was saying earlier it's rare. Okay. All right, these are just 2 slides that show the same concept, but from a different book. And the reason I like this one is you can see that instead of 1 chromosome pair, there's 2. So that chromosome pair versus this chromosome pair, you can see that for this top chromosome pair, it segregates correctly. Okay, and so what they're showing you here in this slide is that a cell only has non-disjunction occur for 1 pair of homologs. Okay. And so in this example, we have non-disjunction occurring during meiosis 2, where in this example, it's occurring in meiosis 1. Okay. I'm sorry, I said that wrong. This is occurring, here's the non-disjunction event. These are both going that way. And this is going from meiosis 1 to meiosis 2. So really, the reason this slide is confusing is up here, this should say meiosis 1. Okay, so during meiosis 1, the non-disjunction occurs. They stick together. And in this slide, okay, oh, there it is. There's the meiosis 1. I don't know why it's missing. I have to fix that. So in this case, non-disjunction occurring during meiosis 2. Okay. The other key difference here is we're not showing the subsequent fertilization in these slides. So in this slide, we see fertilization. In this slide, oops. In this slide, we do not. Okay. So this slide, you can imagine drawing a line here, and then everything above this would represent what's going on here. Okay. All right. So how do we test for this? It turns out there's 4 common ways we can screen a fetus or developing embryo for these situations. Okay. So in addition to blood tests and ultrasound, okay, we can also do what's called amniocentesis or chorionic villus sampling. In amniocentesis, this is a procedure when amniotic fluid is removed from the uterus for testing. And the fluid is basically what surrounds and protects the baby during pregnancy. This fluid contains fetal cells and various proteins. Think of it as a sort of broth that contains lots and lots of different biological markers and living cells. And so what they do is they take this fluid, and in addition to doing a bunch of biochemistry tests on the fluid itself, they take the cells, which are shed from the baby, and they plate them, and they grow them, and they grow them large enough and enough where they can then do a karyotype on them. Okay. And chorionic villus sampling, this is when the this is a little bit more invasive. So the reason we would do this is if the initial tests from, say, blood work or sonography, the ultrasound, results in some kind of significant finding that would possibly impact the management of the pregnancy or the desire to continue the pregnancy. So this is usually done early on in 11 to 14 weeks. And what it ends up doing is basically taking a small bit of the cells on the fetal side of the placenta, and you can immediately do a karyotyping with these cells rather than having to wait several weeks for them to grow up. Okay, and so when I say karyotype, this is what a karyotype is. We talked about these when we did Down syndrome earlier. So a karyotype helps detect very large scale problems with chromosomes. You can't see individual mutations here, obviously. You can't tell if, for example, chromosome 7 is harboring some mutant allele that will result in a disease. But what you can do is you can see whether or not, for example, if you have Cry-do-chat, right? If so, if you had Cry-do-chat, this purple lump here would be gone or cut in half. If you had Down syndrome, you would see 3 copies of chromosome 21. Okay, if you had Klinefelters, you would see multiple copies of the X chromosome. If you were Turner syndrome, you wouldn't detect a Y chromosome or an extra X chromosome, right? So that's what karyotypes do. The other thing you can do is you can detect for chromosomal abnormalities, okay? And this is probably a little trickier, but there are inversions, deletions. Okay, so deletions would be like what happens in Cry-do-chat, okay? Duplications and translocations. We'll go through these. Okay, so which one of these karyotypes is abnormal? And you can see, well, if you didn't read all this, you can immediately see chromosome 21's extra, right? Okay, so the reason why you see these colors is because chromosomes can selectively take up different dyes. That's why we call them chromosomes, right? Colored bodies. And these patterns are very specific to each chromosome, to the point where they are completely recognizable and distinguishable by the trained eye, and they can be put in order and aligned for investigation. Okay, so you can see somebody here working under a microscope looking at a particular karyotype. All right, so as we said earlier, Turner syndrome, Klinefelter, supermales, and we didn't talk much about metafemales or superfemales. Those are individuals that actually inherit a third X chromosome. But these are all the types of aneuploids that can happen in sex chromosomes due to nondisjunction. Okay, all right, now let's talk about these chromosomal abnormalities. So on the top here, you can see that's a deletion. So for example, if this is chromosome 5, you can see that the whole A locus is missing. Okay. In duplications, you can see that during DNA replication, during S phase of the cell cycle, there's a duplication event that leads to a duplication of the DE locus. So you can see here A, B, C, D, E, D, E, F, G. So you can think of this as a genetic stutter. Inversions are when during replication there's an accident that ends up inverting, right? So this could happen during a mistaken crossing over event, or this can occur during DNA repair. There's many different ways that you can lead to an inversion. So instead of A, B, C, D, E, F, G, you get A, B, D, C, E, F, G, right? And so you can see, it's like, imagine cutting this out and then flipping it upside down. Okay, and now you get the D over here and the C over here. So that's called an inversion. And then lastly, we have what's called a translocation. Translocations are like musical chairs, okay? In a way, it's a lot like crossing over, except it's happening between 2 different chromosomes. Okay, so in crossing over, we talked about non-sister chromatids, right? So here's a pair of homologs, right? And so these 2 would swap parts. But the thing is these chromosomes are homologs. Okay, you can imagine another set of homologs where this happens, but instead it happens between completely different chromosomes, and that results in all sorts of bad. Okay, so like if we cut the genes here and then we paste them, so you can see instead of A, B, C, D, E, F, G, H, you get A, B, C, D, E, F, Q, R. Okay, so F and Q are now next to each other when they're not supposed to be. Okay, and actually if you zoom in here, sometimes you get, so if this is the DNA, okay, and let's say this is F, let's say this is F and G. Okay, so this is what it's supposed to look like. You know, F and G, that's the wild type. Okay, and then somewhere else there's P and Q. Okay, there's the P and Q. And so you can imagine if this happens, where these exchange parts, such that you get, sorry for my messy arrows here, such that you get FQ, okay, FQ and PG. If this happens inside of a pair of genes that encode enzymes or protein receptors or any sort of protein, you get what's called a chimeric protein. Chimeric. And a lot of these chimeric proteins, these mutant proteins, can go on to cause different sorts of diseases. Okay, for example, acute promyelocytic leukemia is caused by a translocation resulting in a chimeric protein that causes a leukemia. Okay, so here's just another diagram showing deletions, inversions, duplications, and translocations. Now here's an extra word, reciprocal translocations. Reciprocal translocations mean there's an even exchange of material. Okay, there's no genes getting lost. A non-reciprocal translocation would really be when one of this happens, but the other ends up having a mistake and you don't get the even exchange. Okay, so for example, this chromosome might look like this, but this chromosome instead would look just like this. So you get this entire chunk deleted. Okay. Now what happens when you have an inversion? Okay, so here's a pair of homologs, and you can see at this inversion, what happens is when these two have to line up during meiosis 1, they have to end up doing this convoluted dance so that A is paired with A and B is paired with B and C is paired with C and D is paired with D and E is paired with E and F and G, right? So if these were not, if this did not occur and you had a normal, if this was D and E, okay, oh, and then C is all the way down here, so C was back up here. So there's a whole mess here, okay? This whole chunk was inverted. And so if it wasn't, you would just see normal crossing over, right? But because of this inversion, this chromosome in 3D space has to twist so that it lines up. And when you have these kinds of situations and the crossing over occurs, what this does is that this is how you get a duplication, right? So you can see this whole chunk here, D and E, is now going to go here, resulting in this duplication, okay? And you're going to get a deletion. And the other chromosome, so this chromosome here, you go F, here, the resulting chromosomes would be, let's see, F, G, C, D, E, F, G, right? And so you'd get, here, let me change the color here so you can see better. Okay, so you'd get this chromosome, that's this chromosome, okay, and let's do a different color. Let's do, well, there's not a lot to choose from that aren't confusing here. Let's do black. Oops, I think I picked red. Oh, here, that's fine. We go A, B, C, D, E, A, B. Okay, and so a single simple mutation, this inversion, in one generation of cells, and the next generation can lead to both duplications and deletions, right? And so as time progresses, as generations pass, these chromosomes can get more and more mutated. All right, last but not least, we have the Philadelphia chromosome. The Philadelphia chromosome is a reciprocal translocation between chromosome 9 and 22. So the very tip of the Q arm of chromosome 9 is exchanged with a very large chunk of the Q arm of chromosome 22. So this region of 22 goes here, and this region of 9 goes here, resulting in what you can see. It's not 9 that's the problem, it's 22 that's the problem. That's the Philadelphia chromosome that causes cancer, right? And so what's happening is in here, that's one of those chimeric proteins I talked about earlier that causes cancer. Okay, so that's the end of genetics, and I hope you enjoyed this lecture, and it was not too confusing. This is my first go at making these kinds of video lectures, so I'm sure that I'll be doing this again at some point and fixing it. So by all means, feel free to post some comments and or send me a private email. That's fine. My email, if you want to contact me, is let's see. Let me see if I can do this here. Oops. Let's see. So my email is it's Andrew dot Ippolito at bucks.edu. Okay, so any questions or comments, either post them in YouTube or you can send me an email privately. All right, I appreciate any feedback to make these even better. Thank you and have a good day.